Thank you for the kind introduction. Hello, everyone. Thank you for having me here today. It's a great honor for me to be here, and I'm really excited to present my recent paper that was submitted in Nature Communications called Lateral Hypothalamic Leptin Receptor Neurons Drive Hunger-Gated Food Seeking and Consummatory Behaviors in Male Mice. We all know that when we're hungry, we start eating. And when we do start eating, we go out and seek food, which is called the appetitive phase. And this leads us to discover food, which leads to the consummatory phase. And in the consummatory phase, we eat the food, which leads to the termination of eating behavior. Well, for me, in the morning, I woke up, I was hungry. So I opened the fridge. That's the appetitive behavior. And then I took out some food and then eat the food. That's the consummatory behavior. And when I was full, I terminated the behavior because I had to come to my lab and work. And that's the whole process of eating behavior. Although a lot of people think that eating behavior is just one behavior, you can see in the example and in this review paper that eating behavior is not just one simple behavior. It's separated into the appetitive phase and the consummatory phase. So then we can ask this question, how does the brain encode eating behavior? Where is the eating behavior, the seeking behavior, and the consummatory behavior, where is it encoded in the brain? Are they distinct population in separate regions like the cortex or the midbrain, or are they close to each other, like in the hypothalamus? And in the hypothalamus, are they just close to each other encoding the seeking and consummatory at once, or are they separated again into separate types of neurons encoding seeking or consummatory behaviors? So many behavior neuroscientists try to answer these questions. And we also chose the hypothalamus to investigate this phenomenon. And one of the most popular and well-known population in the hypothalamus, lateral hypothalamus, encoding these behavior is the LH-GABA. They are known to evoke diverse motivation, not just the eating behavior. They encode motivation such as thermal regulation, thirst, eating, sexual behavior, and social behavior. And a recent paper in Stuber's group in 2015, they reported that the LH-GABA neurons are separated into consummatory and appetitive neurons about the eating context. However, it still remains a question into what kind of subpopulation in the LH-GABA neurons are encoding these two separate behaviors. So we thought about this, and then we chose the leptin to research about the leptin receptor neurons because first, they are a lot of leptin receptor neurons, the LEBR neurons in the lateral hypothalamus. And the recent paper told that in the RNA sequencing data, they said that many LEBR neurons are GABAergic neurons, almost a lot of them. And then we used the open data to get the exact number of how much LEBR neurons are GABAergic. And we found that about 80 to 90% of the LEBR neurons are GABAergic neurons. Also, it was known, many research papers told that the LEBR neurons drive eating behavior, like one of the most recent papers said they drive appetitive behavior, but not consummatory behaviors. But the results were very controversial. Some said that activating these neurons decreases consummatory behavior. But some research said instead that activating these neurons increase the consummatory behavior or decrease, and leptin injection into the lateral hypothalamus decrease the food intake. So we thought it would be very interesting to study about the LH-LEBR neurons. So we first investigated if they are the subpopulation of GABAergic neurons that are responding to food-specific context. So to do this, we used the miniscope technique. We injected the GChem6S virus that, when injected into the neurons, they let them create protein that exhibits green light when they meet calcium. Then we implanted the green lens that lets us see this kind of video where we can track all the cells that are activating in the LH-LEBR. So after we got the video, we used the machine learning technique called CNFE to denoise all the signals and extract the true signals for analysis. And then we picked the food-specific neurons according to the scores we defined. And we did the same for the leptin LH-LEBR neurons. So when we got the response, we got the response from a lot of neurons. And as you can see, we define food-specific responsive neurons if they were activated in the 3 food tests and not activated in the non-food tests. So we let the mice do a test where they consecutively do these three kind of tests and then do the non-food test. And as you can see, LH-GABA neurons they had a small proportion of food-specific neurons compared to the LH-LEBR neurons. And we quantified the proportions of these neurons, and as a result, we could see that 8% of the total coverage population for the food-specific responsive neurons were 8%, and we could see that there was 63% of neurons, food-responsive neurons for the LH-LEBR. So we used these facts, and we also knew that from our study and other studies, that there were the leptin receptor neurons and GABA receptor, the GABA neurons that were co-expressing both at the same time. There was about 4 to 20% of them among the LH-GABA neurons. So we thought that about just 10%, if there were 10% of LEBR positive neurons among the LH-GABA neurons, we could get this number, and the 8% are food-specific neurons of the LH-GABA neurons. So we can get this ratio, and then according to this data here, only 63% are the food-specific. So we could get that about 63 out of 80, that's about 79%. We could get that 79% are LH-LEBR neurons are food-specific subpopulations of LH-GABA neurons. And then we also saw if in another context, if these neurons would respond, and we saw in the water context where the mice were dehydrated and they started drinking the water, but we couldn't see that much neurons responding, and the amplitude was also not as high as when the LH-LEBR neurons were responding in the food context. So to summarize, we could see in this experiment that the LH-LEBR neurons are the food-specific subpopulations of LH-GABA neurons. And then we tried to see the LH-LEBR neural activity in the population level using fiber photometry, and we did these 2 tests, the confirmatory behavior test and the seeking behavior test. We injected the GChem6 virus again to the LEBR cream mice, but we inserted a cannula in the LH region this time to collect the light that was coming from the LH-LEBR population. And as you can see in the video, during the confirmatory phase, the neural activity starts to increase, and when they get the food upon food contact, the neural activity increase. And for the seeking behavior, we trained mice in this special maze, and mice usually stays in the shelter, but when they run out of the maze, they travel the maze and get to the end of the maze to get the food. But there's a random shock on the floor, so they just don't come out randomly. They only come to get the food. So we know that when the mice come out, they're coming out because they want to voluntarily seek the food. And as a result, we could see here that as the mice come out, the LH-LEBR neurons activate when they show seeking behavior. And then we asked if the seeking behavior we are observing is indeed because mice are exhibiting the goal-directed behavior towards food. So we conditioned mice in this spatial chamber this time, and in this chamber, the mice was trapped in the shelter and were led to seek food here at the end of this chamber. But on the first day, the mice, as you can see, the mice took a long time to get to the end of the chamber, but at the end of the training day, they got there very fast. So we knew that the mice were well trained. And very interestingly, we could see that when the mice didn't know about the food at the end of the corridor, this is the first day, the neural activity did not increase. But as you can see here, when the mice knew about the food, when they learned about it, the neural activity started to increase when they started to come out. So here we could find that, we could demonstrate that the LH-LEBR activity is indeed because of the goal-directed seeking. And then we saw if the LH-LEBR neural activity would go down when the mice terminated the seeking or consummatory behavior. So as you can see in this video, the mice tries to eat the food, show consummatory behavior, but can't get the food. And so when they terminate, they abandon the food, they terminate, the neural activity goes down. And it's the same for the seeking behavior. The mice were trained to eat the food with the reward cue at the end of the corridor after they turned at the corner. But but on the test day, we took the food out and only left the cue here. So the mice goes to the end of the chamber and look for the food, but finds out it's not there, and they abandon seeking, like this mice here. And at that moment, we could see the neural behavior deactivating. So in the summary, we could see that LH-LEBR neurons are time-locked to seeking and consummatory behaviors. And after we observed the neural activity in the population level, we had two hypotheses. We asked if one individual neuron will activate seeking and consummatory behavior at the same time, or are there two populations encoding seeking and consummatory behavior separately? So in order to find this out, we used the miniscope technique again in this special chamber. This is the movie how we conducted. And in the movie, mice come out to seek the food in the maze during the food trial. And when the food is there, the seeking neurons go up. And when the consummatory behavior starts, it goes down. But the consummatory behavior neurons go up during the consummatory behavior and doesn't go up in the seeking behavior. But in the no food trial, when the mice does the seeking behavior, only the seeking neurons go up, and the consummatory behavior neurons don't activate because there is no food, therefore no reason to exhibit consummatory behaviors. And so to elaborate, we visualized all the LH-LEBR neurons with the miniscope and designed two trials. One is the food trial where there is the seeking behavior, and since there is the food here, we can the mice can show consummatory behaviors. And then we also did the no food trial where mice come out, seek the food, but there's no food here, so mice can't show any consummatory behaviors. And as a result, we could look at a lot of the neural we could see all the neural activity of the LH-LBAR neurons and the seeking neurons activating and consummatory neurons activating. And we got a lot of the neurons and we wanted to define them as seeking or consummatory neurons. So we defined seeking score 1 and 2 and consummatory score and plotted these neural activity onto three-dimensional space. And as you can see, the seeking neurons that were defined had very high seeking scores and low consummatory scores, but the consummatory neurons had high consummatory scores while having low seeking scores. So we could see that about 25% of all the LH-LBAR neurons are seeking neurons and 39% are consummatory neurons. And then we checked if the seeking neurons are indeed responding correctly to our definition. So, and as you can see, we saw that the activity pattern was consistent across the neurons. And we found that they all had similar activity patterns. And during the food trial, they activate at the seeking behavior and decrease at the consummatory behavior. And in the no food trial, they activate during the seeking behavior. And for the consummatory neurons, we got similar results. All the neurons were consistent, activated only during the consummatory phase and not responsive during the no food trial. So here we could find out that two distinct populations of LH-LBAR neurons encode seeking and consummatory behaviors respectively. And then up to now, we only saw how LH-LBAR neurons respond, not how they cause the behavior. So in order to see if these neurons actually cause behavior, we need to artificially manipulate these neurons. So, and if they are, and when we activate these neurons, if the appropriate behavior comes out, then we know that the LH-LBAR neurons have causal effect on these behavior. So in order to do that, we injected channel rhodopsin virus into the LH to first activate these neurons. These virus, when they infect the neurons, they make the channel rhodopsin receptors on the neurons, and when light is delivered to them, to the neurons, the receptor activates all the neurons when they are stimulated with the blue light. So for the test, we let mice do an experiment where they could both show seeking and consummatory behaviors but as a result, we didn't see any change in the seeking behaviors like food zone entry or food contact number, and the food consummatory behavior had no change, like the food intake. So we thought that if we maybe separated the seeking and consummatory behaviors, we might be able to see some change when activating the LH-LBAR neurons. So we first conducted a seeking behavior. This is mice that learned how to seek the hidden food here, but since it's the test state, there's no food here. And as you can see, if only the seeking behaviors are, if we only let the mice do seeking behaviors, you can see the seeking behaviors increasing, like digging for the food, trying to find the food here. Where's the food? I want to eat food. And then, but after the laser is turned off, you can see that the mice stop seeking for the food, it just looks around, and you can see the seeking behavior is decreasing. So to elaborate on how we did this, we first trained mice of this chamber, and where in the four corners there were the food inside here, hidden beneath the bedding. So when the mice were conditioned, they find all the food here and learn how to seek the food. But on the test day, we take the food out, so the mice cannot show consummatory behavior. So, and as you can see, like in the video and in these results, when the LH-LBAR neurons are activated here, the seeking behaviors increase, and we could also significantly show that the seeking like the digging duration, the number of digging increased, as well as the entry to the zone. So here we could see that LH-LEBAR neurons encode seeking behavior. And then we separated the consummatory behaviors, and in here, mice were always had their face in front of the food, so they always could exhibit the consummatory behaviors and not show seeking behavior. And at this time, when we activated, as you can see in the video, the LH-LEBAR neurons make the mice eat food, while the laser is off, the LH-LEBAR neurons don't eat food, don't make the mice eat food. And we could see the results here. When we activated the LH-LEBAR neurons, we could see the increase in consummatory behavior, the number, the duration, and the food intake also increased. And we also did some control experiments compared to the EYFP, and we could see that indeed the activation of these neurons were able to evoke consummatory behavior. And so up to now, all our analysis were done manually by humans. So we thought, could the machines separate these consummatory behaviors? So to do this, we used the deep learning algorithm, now very famous in behavioral neuroscience, called DMLAC-CUT. And as you can see in the video, this algorithm lets us label all the body parts in the mice. And using these labels, we were able to separate consummatory behaviors, like here and here. And we could see that the body parts were far away when the LH-LEBAR neurons were not activated, but when they were activated, very close to the food zone, showing the consummatory behavior. And you can also see the food consummatory duration increasing, and we could significantly see the change when we activated the LH-LEBAR neurons. And then we see if we could, this time, if we inhibit the LH-LEBAR neurons, we could see the decrease in seeking or consummatory behaviors. So this time we inserted the holorhodopsin into the LH-LEBAR pre-mice, and this lets us inhibit the neurons when we give the yellow light. And as a result, we could see that when the mice were doing seeking behavior, we couldn't see the hungry mice showing decrease in seeking behavior. But instead, when we let mice only do the consummatory behavior, we could see that they showed decreased bout duration and decreased consummatory behavior. Like here, you can see that the bout duration is short, but when the laser is off, this means the LH-LEBAR neurons are activated. You can see the consummatory behavior is increasing. And then we quantified the experiment, where in the experiment we inhibited the LH-LEBAR neurons for 2 minutes, and then in a 2-minute interval, and it was on for 2 minutes, off for 2 minutes, and when we quantified the consummatory behaviors, we could see that during the inhibition of LH-LEBAR neurons, the consummatory behaviors were decreased, and we could see that the bout duration decreased when inhibiting the LH-LEBAR neurons. So here we could see that LH-LEBAR neurons are sufficient for seeking behavior, and the LH-LEBAR neurons cause the consummatory behavior. So then we questioned if LH-LEBAR neurons are affected by hunger signals, because of course, as I explained earlier, the reason we start to eat is because we seek for, because we're hungry. And that's why we go to the fridge and take out food and eat. So we thought that one of the most famous hunger signals is MPY, called the neuropeptide Y in our body. So we thought that this would cause this would send the MPY to the LH-LEBAR neurons, and there are a lot of MPY receptors in the LH, and when MPY is delivered to the LH, they evoke eating behavior. So we thought that if we investigated the effect of MPY onto the LH, we could have some good results. And as you can see here, we administered MPY onto the brain slice of LH-LEBR neurons to see if LH-LEBR neurons were activated. And as a result, we could see that the administration of MPY into the LH-LEBR neurons were enough to increase the area under curve or the max activity of these neurons. And we could see that the amount of active cells were also more in compared to when we delivered MPY and the MPY antagonist together. So we knew here that the MPY was activating LH-LEBR neurons, but MPY is actually commonly known to send inhibitory signals because they are the GI protein. So we thought that there might be another mechanism into how these neurons are activating these neurons. So in our scRNA sequence results, we could see that the most of the MPY receptor neurons were GABAergic neurons. So we hypothesized that the MPY GABA neurons they would inhibit the LH-LEBR neurons until the MPY comes and binds to these neurons. So now usually these MPY GABA neurons are inhibiting the seeking and consummatory neurons, but when they bind, they disinhibit the LH-LEBR neurons and they activate the seeking or consummatory neurons. So to check these hypotheses, we used the electrophysiology onto the LH-LEBR neurons. We checked if the inhibitory postsynaptic currents would decrease when we give the MPY to the LH-LEBR neurons, and indeed we saw that the iPSC frequency decreasing when we delivered the MPY to the LH-LEBR neurons. So to summarize all of this, we could see that the LH-LEBR neurons are activated by MPY by the activation of GABAergic interneurons. So this is our model summarizing all our results. We think that when animals are full, when they're in the sated state, the AGRP neurons are not activated, so they don't make the MPY. So the MPY neurons in the LH, they are inhibiting the LH-LEBR neurons. But this means that these neurons are locked, so they keep getting inhibited, so they don't increase even when we are in front of food. But when we're hungry, the MPY comes out and they bind to these MPY GABAergic neurons, and they disinhibit the MPY neurons to make ready to evoke the eating behaviors. This means that the lock is open and the LH-LEBR neurons are ready to evoke the eating behavior. And then when the animal or us, when we're in the situation to eat, these neurons activate and they orchestrate, they activate in this sequence, make the seeking behavior come out, and then let the consummatory behavior come, and they do these behaviors to recover a homeostasis for survival. And now I would like to give my thanks to to the researchers here, Young-hee Lee, Yubin Kim, and Mire Zhang. They are the first co-authors who did this research with me, and I give thanks to the two corresponding authors, Sang-jung Kim, and especially my PI, Hyung-jin Choi, who made all the work possible. And I thank the rest of the authors who contributed to this paper and all our lab members who gave a very constructive and inspiring comments during our research. Thank you, and I'll get the questions now.